Okay, we're on the last chapter. We're going to be talking about amphibian and reptilian and actually Chelonian anatomy and physiology. Let's get started with our learning objectives. We're going to list the taxonomic orders in the classes Reptilia and Amphibia. We'll define ectothermic and explain how ectothermic animals regulate their body temperatures. We'll list the unique features of the reptilian and amphibian integumentary systems. We'll describe the process of ecdysis. What is ecdysis? That's really exciting. You're going to learn about ecdysis today. List the unique features of reptilian and amphibian vision and hearing. We'll list the components of the reptilian and amphibian heart and describe the flow of blood through the heart. We'll describe the unique features of the reptilian and amphibian respiratory and gastrointestinal systems, describe the structure of the kidneys of reptiles and amphibians, and describe the factors that determine the sex of offspring of reptiles and amphibians. It's pretty cool. We're going to list the unique features of the musculoskeletal system of reptiles and amphibians as well. So in the taxonomy, in the class Reptilia, there are 4 orders. Crocodilia, which are alligators and crocodiles, Squamata, which are snakes and lizards, Chelonian, which are turtles and tortoises, and Rhynchocephalia, which are totteras. And these are pretty rare lizard-type creatures. They're not lizards. They're Rhynchocephalia. They look like this. They kind of look like iguanas, but these are Rhynchocephalia. They're totteras. All right. In the class Amphibia, we have 3 orders. Gymnophiona, which are caecilians. These are caecilians here. They are blind large worm-like creatures. You can see they have a mouth and nostrils, and the eye is covered with skin. We also have Anura. Anura are frogs and toads. And Caudata are salamanders and newts. Metabolism. These are ectothermic animals, and they are unable to generate body heat internally. Their body temperature depends on their environmental temperature. So what do we commonly call that? They are cold-blooded animals. Some reptiles can actually raise their body temperatures via metabolic processes, for example, through muscular contractions. So there are some reptiles that can adjust their body temperatures internally, but it's pretty rare. Most of them are, for the majority, cold-blooded. Herptiles maintain body temperatures via behavioral thermoregulation, so postural changes, meaning they change where their body position is in relation to the heat source or the cold source. Ectothermy is related to energy conservation. An animal regulates the temperature depending on its metabolic needs. This allows many herptiles to survive on very small amounts of food. So if they are not able to get food, they can go into a colder environment, will slow down their metabolism, and they don't need food. So this depends on access to temperatures within the preferred optimal temperature zone. The POTZ is the range of temperatures in which the animal can perform all necessary metabolic functions. So they need a cold spot or cooler spot and warmer spots. And it's really important that your clients understand what the POTZ is of the animal that they are housing. Their skin, they have a keratinized skin. There is alpha keratin, which is soft and flexible, and it's found in the interscalar skin, so between the scales. Beta keratin is rigid, and that's the keratin that is found in the scales. They have very limited subcutaneous space, so we don't give a lot of sub-Q injections into these guys. Most of the injections we give to reptiles are IV, intraperitoneal, or IM. So this is the epidermis, where the keratinized skin. We have our small amount of dermis, and then we have our muscle tissue or bone tissue here. There are melanophores in the skin. These are pigmented cells. The osteoderm, which is a bony part, and here's our flexible hinge made of alpha keratin. Right, so within the dermis, we have dense connective tissue. That's where blood, lymph vessels, nerves, and chromatophores, which are also those pigment-containing cells. Chromatophores will allow some lizards to change skin color and pattern. osteoderms are these bony plates within the dermis of some lizards and crocodilians. Integument. Scales are formed by epidermal folds in most reptiles, and they vary in size and shape. There is a spectacle, there's a special modified scale called the brill that may cover the eyes. And you can see, you can't see it here, but you can see it's cloudy here because that's right before a shed, and this is after a shed. But that's an actual scale or spectacle or brill over the eye. Here we have an cell here. We have our mantle muscle, some fluid, blood vessels coming in here, nerves. These are, this is actually an eye with the lens, some reflectors, some muscles, and skin with chromatophores here. There may be other epidermal structures that are present, such as crests along the back, tubercles, these are tubercles here, spines, dewlaps. This is a big dewlap here, so there are other epidermal structures on reptiles. We have scale and scute nomenclature, which aids in species identification and medical recording. So this is the shell of a Chelonian. We have a cervical scale, and 1, 2, 3, 4, 5, and we have our pleural scales and our marginal scales. So cervical, vertebral, pleural, and marginal scales. Ecdysis, very exciting. It's the shedding of the skin, and it occurs with growth and in response to skin injury. Its process is controlled by the thyroid gland. It's shed in pieces or in one large piece, like this animal here. This is a coral snake. This is a shed skin, okay, and that is called the exuvia. The exuvia is the product of ecdysis. Cells replicate a new epidermis. We know that about our own skin. This happens in snakes as well. There's an enzyme containing lymph that is secreted between the old and new epidermal layers. The skin color dulls, and then that spectacle opacifies. So when you see a cloudy brill or a cloudy eye, that's when we know that a shed is about to happen. There's actually fluid underneath that scale. There's lymph, that's the fluid that is, and it's resorbed prior to ecdysis. So when they get that separation, and that fluid is in there to separate the old from the new, and then it's resorbed, and then we have this dry skin on top that is removed by mechanically rubbing on objects. Amphibians have a slightly different skin. Their epidermis is a single or few layers of keratinized cells, but aquatic amphibians have no keratinized cells. So they have very thin and skin that can be damaged very easily. It's extremely permeable. They actually absorb water directly from their environment. They don't drink it. They absorb it in their skin. There are drink patches, which are areas of increased permeability on the ventral surfaces. The dermis itself is where the chromatophores and glands are, and those glands produce secretions which help protect the amphibian skin. So here are some parotid glands on this toad here. Now, if you've ever had a dog who's picked up a toad, you'll know that they start to froth at the mouth, and that the reason is that those glands are producing secretions on top of the amphibian skin to make it bitter tasting, and that bitter taste creates a lot of saliva in the dog's mouth. They drop the toad because it tastes horrible, and the toad hops away. They also can have toxic secretions by some glands within the dermis and the epidermis as part of their defense mechanisms. In fact, there are certain salamanders that can create a toxin that induces seizures. So sometimes We would see this up in New Hampshire quite a bit in the springtime when the salamanders were coming out as they sought mates or places to lay their eggs. We would see increases in seizures in dogs that were out hunting salamanders. Their dermis underneath the epidermis is, again, very little subcutaneous space in salamanders and caecilians. And urines, frogs and toads, have looser attachments of the dermis, so they do have a little bit more subcutaneous space. Amphibians regularly shed outer layers of the epidermis, much like we do. Vision. Reptiles, the iris is made up of skeletal muscle under voluntary control. Wouldn't that be cool for us to be able to do? We can't do that. Ours is under involuntary control. They do have a pupillary light reflex, so they do have an involuntary control, but they can also control that iris, open it up, close it down. They don't have consensual reflexes with their pupillary light reflex, so if we shine a light in one eye, the other eye does not constrict as well. Their lower lid is usually more mobile than their upper, so when they close their eyes, it's that lower lid that closes, not the upper lid. Vision for these guys, some species of lizards have thin, transparent lower lids that allows for a degree of vision even when the lids are closed. There are these things that are cartilaginous pads. They're called tarsal pads. When we see tarsal, we often think of the back foot, but in this case, these are pads that are found in the lids of some reptiles, and they produce a certain type of tear, a mucousy type of tear. The nictitans, or third eyelid, is well developed and mobile in many of these reptiles. Snakes and some lizards have no true eyelids. They have that clear fused scale, which is the spectacle or the brill. There is a tear film that is between the cornea and the spectacle in the subspectacular space. Subspectacular, that's a great, actually, it's a pretty spectacular word, but that subspectacular word is under the spectacle, and there's a tear film between that to protect the cornea. Tears then drain into the mouth through the nasolacrimal duct system. Hypovitaminosis A, this is why this is important to understand. Hypovitaminosis A, when we have not enough vitamin A in the diet, leads to abnormal epithelial cell growth. It will have flat, abnormal functionality of those epithelial cells, which will lead to squamous metaplasia, particularly of the lacrimal and Herrerian glands, and that causes eye issues. So this is a turtle with hypovitaminosis A, and it causes swelling of the epithelium around the eyes. And that's a normal turtle and a turtle with hypovitaminosis A. So this is why that is important to understand. Most reptiles have poorly developed extraocular muscles, with one exception. Chameleons, as you know, their eyes move freely and independently of one another. Chelonians also have no nasolacrimal duct, so their tears spill over the lid margin. They're always Chelonians, not chameleons. Chelonians are always crying. Those tears spill over the lid margin. Turtles, when you see turtles tearing up, that's normal. Lacrimal and Harderian or Herrerian, I believe it's supposed to be Harderian glands, are present in most reptiles and amphibians. They produce secretions that combine to form the tear film. Eyelids are absent in some aquatic species. Caecilians, like I said, they're blind. Their eyes are actually covered with skin. And with amphibian eyes, they often protrude ventrally into the oral cavity when the animal is swallowing. So think about if you dissected a frog in high school biology. When you opened up the mouth, you could actually see the eyes protruding into the oral cavity. All right. These guys also have ossicles or scleral bones present in most reptiles, just like birds, except with snakes and crocodilians. Snakes and crocodilians don't have that sclerotic ring. The lens is more fluid in reptiles than in mammals, except in snakes. It's more rigid in snakes. It's really cool with accommodation of the lens, which means that it's contracting or it's either getting bigger or rounder to focus on what they're trying to focus on. With turtles and tortoises, that lens is actually squeezed through the pupil. You see a picture of that here. That's normal in a turtle or a tortoise. If we were to see this in a dog or a cat, we would freak out because it would mean that that lens is subluxated, and that would be a problem. With snakes, the lens moves back and forth due to pressure changes within the aqueous and vitreous humors. So it's not really controlled necessarily by the muscles or the ciliary bodies. It's actually controlled by the pressure changes within the eye. Reptiles have avascular retinas, meaning there's no blood supplied to the retinas. Nutrition and waste removal is via choroidal vessels in the vitreous, so it's through the vitreous humor. The conus papillaris, so a little finger, a little cone, in lizards, it actually extends into the vitreous from the optic disc. It provides nutrition and waste removal. The crocodilians have a tapetum, so if we shine a light and we can have the eye shine in the back, the tapetum in the back of the eye, that reflective surface on the retina shines back at us. So that's one way of finding alligators and crocodiles if you were happening to go out and hunt them at night. A parietal eye, or a third eye, is found in some reptiles. It's has a rudimentary retina and cornea with no iris, lids, or musculature. We don't know what that function is. That parietal eye is found up on the top of the skull. Really interesting. It may play a role in light cycle mediated hormone function, but its exact function is unknown. For the cardiovascular system, the location of the heart will vary based on what species it is. Chelonians, or turtles and tortoises, their heart is on the midline, just caudal to the thoracic girdle and ventral to the lungs. Most lizards, the heart is within the thoracic girdle. With crocodilians and some lizards, it's farther back in the coelomic cavity. Now again, there's not a separation or a diaphragm between the chest and the abdomen, so we call that the coelom, or the coelomic cavity. And it's back towards the stomach a little bit. In snakes, the heart is usually at the junction of the first and second third of the body length. So if you drew the body of the snake, you would find it, and you divided it into thirds, you would find it between the first and second third of the body length. It's actually fairly mobile within the coelomic cavity, and it will adjust its space within that coelomic cavity depending on what's going on with the digestive tract. The heart, there are 2 atria and 1 ventricle. With crocodilians, they actually have a 4-chambered heart with 2 aortas, right from the left ventricle, and the left one comes from the right ventricle, and they're connected near the base of the heart by the foramen of Panizza, which allows bypassing of the lungs when necessary. So there's a lot of mixing of oxygenated and deoxygenated blood within the heart. Ventricle regions, there's the cavum venosum, which is where the paired aortic arches come out, leading to systemic circulation. There's the cavum arteriosum, which receives blood from the pulmonary veins and directs the oxygenated blood into the cavum venosum. And we have the cavum pulmonale, which receives blood from the right atrium and directs flow into the pulmonary circulation. So a lot of mixing of bloods. Can shunt past the lungs if necessary if the animal is not using its lungs at the time. The heart, the pressure differences of the outflow tracts and the muscular ridge that partially separates the cavum venosum from the cavum pulmonale maintains some separation of oxygenated and deoxygenated blood. It's just a little bit more complicated because it's just a different structure than we're used to seeing. Now, we can decide what is a normal heart rate based on it's based on species, the size of the animal, the temperature. If it's a cold environment, their heart rate's going to be lower, their activity level, and their metabolic function. Their heart rate is based, like I said, on a mathematical formula, which is 33.4, a constant, times their weight in kilograms to the negative 0.25. It's not important necessarily that you remember this calculation, but you should know where to look for it if you are doing a lot of work with reptiles. Lizards have a vasovagal reflex, which induces a drop in heart rate, blood pressure, and puts them into a catatonic state. We can actually trigger this vasovagal reflex by applying gentle pressure to both eyeballs through the closed lids. Once we remove the pressure or we give them a mild stimulation, they will recover. But this helps us when we're restraining a lizard to keep them nice and quiet. Their blood cells look like avian blood cells. Their red blood cell is oval and nucleated. They have a really long lifespan, between 600 and 800 days. For several years, this red blood cell can live within the body. And that just has to do with the fact that they have a very low metabolic rate. Immature erythrocytes can occasionally be seen, usually in juveniles and during ecdysis. So if we take a blood sample, we see some immature erythrocytes, we can tell that the animal is older. They're probably going through a shed. The heterophyll is round with eosinophilic or red rod-shaped granules and round to oval nuclei. It's analogous to the mammalian neutrophils, but it does not have peroxidase and acid phosphatase. Reptiles produce caseous pus instead of liquid material. So when we have a lot of neutrophils in one place in a mammal, we see pus or kind of a liquid material. With reptiles, we have cottage cheese material. It's very thick, doesn't have a lot of fluid in it. Eosinophils are similar in appearance to heterophiles, but the granules are round. So here's a heterophyll, rod-shaped red granules. Here's an eosinophil. We have round granules. Basophils are small round cells with deeply basophilic cytoplasmic granules, and that may obscure the nucleus. So you can barely see the nucleus here. And here are platelets. These platelets are nucleated, which is different from what we would see with a mammal. Blood collection. How do we get blood? We get it through cardiac puncture. So that makes it really important to understand where the heart is in a snake. And look, here is a snake. The head is up here. Tail is down here. This is the first third of the snake. This is the second third of the snake. And there's the heart. Tail vein is very commonly used. The ventral abdominal vein can be used. This is a sea turtle. This is a jugular vein on a sea turtle. The coccygeal vein, which is the tail. The brachial vein, which is the front leg. And the post-occipital sinus. So behind the cranium, typically of a Chelonian, is a place that we could get it as well. More blood cells. Lymphocytes vary in size. They're usually round cells with large nuclei. They're not really, this one is not really round. They have a large nuclear to cytoplasm ratio and no cytoplasmic granules. So they're agranulated, just like in mammals. Monocytes are oval or lobed nuclei, blue-gray cytoplasm. They may have some small vacuoles or very, very fine granules within them. Some reptile monocytes actually have small azurophilic granules, azurophils. Thrombocytes are small oval nucleated cells with a colorless cytoplasm that may contain some small granules. The respiratory system in reptiles, because reptiles are capable of surviving long periods without breathing, they have a large pulmonary volume, and they have very efficient anaerobic metabolism and cardiac shunting capability. So we talked a little bit about that in the cardiac section, where depending on the pressure, blood pressure within the system, it will change where the blood goes. Respiration is driven by oxygen levels in the blood. So when oxygen gets too low, then they will breathe. The glottis is really easy to find in amphibians and reptiles. It's right at the base of the tongue, actually in the rostral portion of the oral cavity. Here's this big hole here. So it's really easy to tube a snake or a reptile. Glottises are very mobile in snakes. They protrude from the mouth. They can protrude from the mouth to allow respiration during ingestion of prey. So this guy can breathe even if his mouth is full. There are paired arytenoid cartilages on either side that border the glottal opening, and they are open during respiration, just like in mammals. They do not have vocal cords The only vocalizations that are possible are hissing, grunting, and bellowing. Frogs and toads have vocal sacs that rise from their trachea, and you can hear frogs and toads making certain chirping sounds or singing sounds or bellows. There is a glottal keel that's present in some species of snakes, which increases the volume of their vocalizations. So this keel is right in the center of this glottis, so that when air goes past it, it does make a larger sound. This is the lung of a snake. Here's the trachea that is going through it. It has a honeycomb appearance, and the openings of the honeycomb end at faveoli, not alveoli, but faveoli. These are fixed structures surrounded by capillaries, and this is where gas is exchanged. So these lungs, again, are not as flexible as mammalian lungs, but they're more similar to avian lungs. The tracheal rings are incomplete, except in Chelonians. Turtles and tortoises have complete tracheal rings, like our trachea. There are different types of lungs. This is a unicameral lung, so one sac. It's a simple sac-like lung. It has a cranial portion where the site of gas exchange is. The caudal portion is avascular, and it is kind of like an avian air sac. This is a multicameral lung. It has many compartments with intrapulmonary bronchi. So there's a lung section and then several sac-like structures. This is a paucicameral lung. It has characteristics of both unicameral and multicameral lungs. Reptiles do not have true diaphragms. The action of intercostal, so in between the ribs, intercostal muscles and parts of the axial musculature are used for respiration. Crocodilians do have a muscular septum that is caudal to the lungs. The cranial aspect of the liver is attached to that septum. And it's also the cranial aspect is attached to the septum. The caudal aspect of the liver is attached to the pubis by the diaphragmaticus muscle. Lung inflation actually results from contraction of that diaphragmaticus moving the septum caudally. So it actually moves the liver and the septum caudally. Amphibians have simple sac-like lungs. Some salamanders have no lungs. They actually breathe through their skin. It's called cutaneous respiration. Pulmonary ventilation results from pumping of the buccal cavity or the cheek cavities and the pharynx. Gas exchange can also occur across mucous membranes of the buccal cavity, the pharynx, and the cloaca. They're butt breathers. Ears, hearing. Ears are on both sides of the head, usually caudal to the eyes. They do have a tympanum, which may lie in a depression or be covered by folds of skin in some lizards and crocodilians. They have a columella, much like the avian, which is a single bone in the middle of the ear of reptiles. It connects to the tympanum, which is the eardrum, and the quadrate bone, which is a part of the skull. This transmits vibrations that the skull feels to the oval window of the cochlea. And that's converted to nerve impulses that are then transmitted to the brain via the vestibulocochlear nerve, just like in mammals. Now, what happens is if the snake or lizard has their head on the ground, they can feel those vibrations of you walking up to them. And so that's how they know that you're there. They do have semicircular canals to control balance and equilibrium in most reptiles. Snakes have no external ears. The columella articulates just with the quadrate bone and allows snakes to be very sensitive to ground vibrations transmitted through the mandibles. Snakes are also able to hear aerial sounds, so they can hear when there is sound through the air, but it has to hit the vibration of the quadrate bone. Salamanders and caecilians have no tympanic membranes. Columella actually may be degenerate, so salamanders and caecilians do not hear very well. Feeding strategies. There are carnivorous, omnivorous, and herbivorous reptiles. They all exist. Snakes, crocodilians, and adult amphibians are strict carnivores. They can eat nothing but meat. Some diets are actually very specialized, so it's really important that your client understands the animal that they have and what is appropriate for them to eat. Snakes and lizards have deeply forked tongues. They function as a particle delivery system for the vomeronasal organ, or the accessory olfactory organ. They allow for detection of particle gradients. So here is Jacobson's organ, which is the vomeronasal organ, and the tongue flicks out, picks up little particles, and brings it past this organ, which then senses and detects those particles. Chameleons have a specialized projectile tongue that's designed for capturing prey at long distances. So here is a chameleon capturing prey. There's a sticky end to their tongue, and that's what sticks to the prey item. Turtles and tortoises have a thick, fleshy, relatively immobile tongue. So they don't use their tongue very much as they are gathering food. They use their beak. Crocodilians have an immobile tongue that's attached to the intermandibular space with muscular flaps from the base of the tongue and dorsal pharynx, which allow for opening of the mouth while they're submerged without ingesting or inhaling water. So they can close off their glottis and their esophagus completely with their tongue. The tongue is used to capture prey for most amphibians in urines. Most terrestrial salamanders and caudodorsal that caudodorsal aspect of the tongue is flipped, and the is flipped cranioventrally to prehend food, and that's called lingual flipping. So that basically it's folded over, they bring it out, flip it, and bring it back, and capture it and bring it back. Here's those big parotid glands. This looks like an evil monster frog. All right, there are numerous salivary glands. These salivary secretions provide lubrication that aid in the ingestion of large prey. They also have some enzymatic properties, so digestion is occurring in the mouth. Venom glands with some snakes and lizards are a modified salivary gland. And you can see that this animal has fangs that come out, and they are provided venom through this salivary gland. Dentition. Turtles and tortoises have no teeth. Okay, this is photoshopped right here. So that's an evil tortoise, but not really. What they have are keratinized beaks, or tomia. Other three reptiles, with the other three reptiles, there are actually three types of dentition. There's thecodont dentition, where the teeth arise from sockets in the skull bones, and this we see with crocodilians. So the teeth come from sockets within the skull bones. That's thecodont. Pleurodont dentition is when the teeth are attached to the medial aspect of the periosteum on the mandibles and maxilla, and that's snakes and iguanas, or iguanid lizards. So they are pleurodont. Acrodont dentition is when the teeth are fused to the biting edge of the mandible and the maxilla, and that we see in some lizards. So acrodont, they are fused to the bone. Pleurodont, they're attached to the periosteum, or the coating, or the fibrous coating over the bone. Acrodont, they're fused. I'm sorry, thecodont, they come from sockets. Snakes have 6 rows of teeth, 2 mandibular, 2 maxillary, and 2 on the palatine pterygoid bones. 6 rows. There are aglyphous snakes that lack fangs. So not real fangs, but some canines. And those are mainly snakes that will squeeze their prey to kill them and then swallow them whole. Venomous snakes have very specialized dentition for venom delivery. There are proteroglyphous and opisthoglifous fangs. These are fixed upright fangs. So proteroglyph are fixed fangs. Selenoglyphous fangs are folded on the roof of the mouth when the mouth is closed, and are erect when the snake bites. So selenoglyph are hinged fangs. So they're closed when the mouth is closed. They're folded over, and when the mouth opens up, they hinge into place. So proteroglyph, the difference between proteroglyph and epistoglyph is that these are fixed in a forward facing projection, and these pistoglyph fangs are rear fanged, so they're fixed in a they're pointing caudally. The venom delivering teeth are hollow with an opening near the end where the venom is expelled. The fangs receive venom through a duct with venom gland at its base. Contraction of muscles around the venom gland forces venom out through the fang, and snakes can control the amount of venom delivered with each bite. So if they're biting a small prey, they don't need to use a lot of venom. Most amphibians have teeth. Caecilians and salamanders have both maxillary and mandibular teeth. They may also have palatal teeth in some species. The maxillary dentition is present in some anurine species. They also have odontoid processes, which are cutting plates on the rostral mandibles of some frogs. That's probably something you never thought of, that these little guys have teeth. All right, digestive system. Further on, we're talking about the esophagus. The reptile has a thin and very distensible esophagus. There's unique morphology to each species. It just reflects what type of prey they may be eating. The amphibian esophagus is very short and wide, especially in anurines, toads or frogs. So here, excuse me, is the esophagus and the Chelonian going into the stomach. Here's the trachea, which is much longer and curved, going into the lungs. In the stomach, the reptile has variable size and shape of the stomach. Snakes have a highly distensible stomach. Obviously, they're able to eat prey that is considerably thicker than they are. Crocodilians, their stomach is a thick muscle and we compare that to the avian gizzard. So they can really grind up those big bits of meat. Amphibians, anurines, are actually capable of prolapsing their stomach through the mouth. That's a bad thing. When you see that gastric prolapse, it's a terminal event in a dying animal. So you do not want to see that. When we used to see that when I worked in Banfield at PetSmart, I knew that the best thing we could do was to euthanize this animal. The amphibian intestinal tract, there are indistinct regions. So there's the stomach, and then there's the intestine, and there's the colon. So we can't really distinguish between those different regions. The liver may contain melanomacrophages, which are involved in the immune function. So it's just a different cell, a pigmented cell that eats things. The pancreas is between the stomach and the proximal segments of the intestine, and it does the things that pancreas are supposed to do. The intestinal tract will vary according to the diet. In herbivores, they have longer intestinal tracts than carnivores because it takes more time to digest vegetable matter. Snakes have a relatively straight intestinal tract, which makes sense because they are relatively straight. The colon is large and complex in herbivores because they do use hindgut fermentation for digestion. Ceca or cecum, this is the site of hindgut fermentation in herbivorous lizards and Chelonians. The liver is usually large and bilobed, much like an avian. The cloaca is a common outflow tract for GI and urogenital tracts in all reptiles and amphibians, and is just like avian. There are three chambers. There's the coprodium, the urodium, and the proctodium. Reptile kidney is usually oblong and smooth surfaced. In snakes, their kidneys are lobulated. So this is a snake kidney. The ureters empty into the dorsolateral aspects of the urodium. Some species do not have urinary bladders. Most snakes do not have urinary bladders. There's no distinct renal pelvis, just like in avians. The distal collecting tubules join into collecting ducts that merge to form the ureter. There is no loop of Henle present, so they are not able to concentrate urine as animals do. Water can be absorbed from urine through the wall of the urinary bladder if they have it, from the rectum, or from the cloaca. And so when we mix all of these things together in the cloaca, it allows urine to go into the colon or the rectum so that it can be reabsorbed into the body if need be. Because they don't concentrate urine within their kidneys, sometimes they have to reabsorb water in a different area. Nephrostomes. Nephrostomes connect the coelomic cavity to the renal tubules and filter blood and coelomic fluid. The kidneys are usually lobulated and in the caudodorsal coelomic cavity. So within the amphibian kidney, they have nephrostomes, which are a little bit different from what we see with mammals and other reptiles. Some amphibians excrete ammonia as nitrogenous waste. Others excrete urea, and some excrete uric acid. Do you remember what an avian will excrete as the nitrogenous waste? It's uric acid. Amphibians also have no ability to concentrate urine. Urinary bladders and cloacal anatomy is very similar to reptiles. Reproductively, males have internal testes, just like avian, and they're located in the dorsal coelomic cavity near the kidneys. The ductus deferens or vas deferens leads from the testes to the dorsal wall of the urodeum. Most also have a sexual portion to the kidney tubules. This develops in response to high levels of circulating sex hormones, and they probably provide secretions that contribute to the seminal fluid, so just like the prostate and the vulvourethral glands. The copulatory organs will vary in structure. Crocodilians and chelonians have a phallus of erectile tissue that arises from the floor of the cloaca. Snakes and lizards have paired hemipenes that are everted from the tail base through the vent. So they both come in crocodilians and chelonians. The erectile tissue comes from the cloaca, and in snakes and lizards, the hemipenes come into the cloaca, but exit closer to the vent. Females have paired ovaries in the dorsal coelomic cavity. These paired oviducts lead to the cloaca and end at the genital papilla in the dorsal wall of the urodeum. So they have ovaries on both sides, whereas the avian has it on the left side. The oviduct has 5 regions, just like the avian. Infundibulum, which captures the egg, the magnum, the isthmus, the uterus, and the vagina. The albumin and shell in oviparous species are added to the ova in the oviduct prior to egg laying, so oviparous species lay eggs. In viviparous species, which are live births, the fetuses are retained in the uterine portion of the oviduct. Vitellogenesis is the development and maturation of follicles. This is triggered by environmental cues that stimulate the release of FSH and estrogen. Vitellogenin is a lipid substance which is added, so a fatty substance added to the yolk within the developing follicle. Large amounts of calcium are also added. In viviparous reptiles, there is some support of the fetus. Some lizards actually have a true placenta, more like a mammal. Oviposition is laying of eggs. Many reptiles dig nests to lay eggs, and without suitable nesting material, the female may experience dystocia. Chelonians and lizard species may excavate deep holes to deposit eggs. This is a sea turtle laying her eggs. After oviposition or parturition, reptiles are not involved with care of their youngs. Eggs or offspring. There are some exceptions. Crocodilians protect their nests and young for a period of time following hatching. Pythons and cobras protect their nests until hatching. So this is a little crocodilian hanging out in mama's mouth. Egg incubation. Development and hatching requires proper temperature, humidity, and gas composition of the nest. Incubation time and temperature varies depending on species. Reptile eggs, in contrast to avian eggs, should not be rotated during incubation. You just want to put them in and leave them sit and just keep them in the right environment. Sex determination in reptiles. This is really cool. It's determined by either genotype, like we would determine female XX or male XY, or it's determined by temperature at which eggs are incubated. The sex chromosomes in reptiles are ZW, which is a female. That's heterozygous. It's a ZW. And males are homozygous, ZZ, so it's different from mammals. Here is what indicates the sex ratio, males to females, based on temperature. With this snapping turtle, if the temperature is between 24 degrees and 28 degrees Celsius, it is more likely to have male versus female. With a Trachema scripta, a tortoise, we would have more likely 100% male between temperatures 26 and 29 degrees Celsius or 28 degrees Celsius. It would be 100% male. As it gets colder, it's more likely to be female. And again, we have another bell curve with the alligator. Some species do not have sex chromosomes, and in this case, we have higher incubation temperatures usually produce males in crocodilians and lizards, and the opposite occurs in chelonians. So lower temperatures produce males in chelonians. That temperature range within the nest allows for production of a mixed clutch of hatchlings. So a little closer to the surface, it's a little bit warmer, you'll have males. A little bit down further in the ground, or I'm sorry, you'll have females if it's a little bit warmer, and if it's a little bit colder for turtles deeper in the nest, will be males. Snakes, there is no real sexual dimorphism, meaning you can't tell a snake is male or female by looking at it. You actually need to see evert the hemipenes to determine if it is a male. But in some species, they can have pelvic spurs that are larger in males than in females. These are pelvic spurs here. They are remnants of the pelvic limbs, and they provide tactile stimulation for females when they are mating. Male chelonians have a concave plastron and a more distally located vent, which allows closer apposition of the cloaca when the male mounts the female. So they have a concave plastron which fits over the female's shell. Some lizards do show some obvious dimorphism. For example, the male Jackson's chameleon has 3 well-developed horns on its face. So this is a male chameleon. Amphibians, there is some sexual dimorphism in some species. The poison dart frog males have very enlarged toe pads to grasp the female, and have some male lemurians have very large tympanic membranes because they need to hear calling from other anurans. There are prominent cloacal glands in male salamanders. So this is the underside of a male salamander with prominent cloacal glands. Paired gonads, the amphibians have paired gonads in the dorsocaudal coelomic cavity. Bitter's organs are ovarian remnants near testes in male toads. So toads can be born male or female. If they're born male, they actually have ovarian remnants. So they have both sexual organs, and the ovarian remnants in this case are called Bitter's organs. Some caecilians are viviparous, so they have live births. Most anurans and salamanders are oviparous. There's something called amplexus, where male frogs and toads fertilize the eggs as they are laid while grasping the female. So amplexus, they're not actually fertilizing internally, they're fertilizing the eggs while the eggs are being laid by the female. A phallodium is a portion of the cloaca in caecilians that is everted to deposit semen into the female's cloaca. So they do have intromission. Caecilians have intromission. Spermatophores are little packets of sperm that are deposited onto a substrate like this stick by male salamanders. And then the female will walk over this sperm, these little packets of sperm, and it will be picked up by their cloaca. A spermatotheca is a pocket in the cloaca of female salamanders where sperm is stored. So they'll walk over this sperm, store the sperm until they're fertile, and then will fertilize those eggs. With amphibians, their eggs are usually deposited in or near water. There is some parental care of young eggs and or young in some anurans and most salamanders. Larval anurans or tadpoles have completely aquatic lives prior to metamorphosis. Tadpole metamorphosis is usually complete within 90 days, and metamorphosis is stimulated by thyroid hormones. Now remember, the thyroid controls growth and metabolism, so it makes sense that the thyroid in reptiles is in charge of ecdysis and metamorphosis. The endocrine system. There's a single thyroid gland except in the lizard. With crocodilians, they actually have bilobed thyroid with a thin isthmus between the lobes. The size of the thyroid gland varies according to season and metabolic state. So when it's needed, it is bigger. Parathyroid glands and ultimabrachial bodies are in the cervical region. Adrenal glands are usually within the ligaments that suspend the gonads. They produce epinephrine, norepinephrine, aldosterone, and corticosteroid, just like our adrenal glands do. There's no distinct separation of the adrenal cortex and medulla. The exact location of the endocrine organs in amphibians varies. The nervous system. There are three major divisions of the reptilian brain. There's the forebrain, which has the olfactory lobes, the cerebral hemispheres, and the diencephalon. There's the midbrain, which has the optic lobes, the cerebral peduncles, and the nerve fibers connecting the forebrain and the hindbrain. And then the hindbrain has the cerebellum and the medulla oblongata. There's generally no surface gyri or sulci on the brain. That means there's not a higher level of thought. So the smoother it is, the less thought is higher levels of thinking are going on. So here's a reptile or alligator, really, really big olfactory bulb and a very, very small cerebrum. In a bird or a goose, there is a little sulcus here, so there's some thinking going on here. Here is a rat, which we know rats are fairly smart. They actually have a bilobed brain. And then in a human mammal, we have lots of gyri and sulci. Brain meninges, they have a pia arachnoid layer and a dura mater, very similar to our meninges. They also have 12 cranial nerves, just like us, and they depend on spinal segmental reflexes and locomotor centers for control of movement. In amphibians, their brain is well developed for basic functions, so sight, olfaction, and movement. They only have 10 cranial nerves, and their spinal cord extends to the very tip of the tail in amphibians and caecilians, but ends in the lumbar region for frogs and toads. Animal is dependent on the spinal segmental reflexes to control movement. Their skulls, there are two types of skulls that we can see within reptiles. Anapsid means no temporal openings, and we see this in Chelonians. So an anapsid has no openings on the side of the skull. With diapsid, there are temporal openings in the skull. Snake skulls are extremely mobile. They have a mandibular symphysis that is connected by ligaments that allow the jaws, one side of their left or right, to move independently of each other. That mandibular symphysis is connected by ligaments, and it allows those jaws to move independently, so the right side can go down, the left side can go up. They also have these quadrate bones that allow wider opening of the mouth. So this hinge is like a double hinge. Here's their quadrate bone. Remember, the columella is attached to that, and that's how they hear. We have our squamosal bone on the top part of the skull and our mandible on the bottom part of the skull. Here's the brain case, pretty small, right? So quadrate bone is this hinge that allows this top part of the skull to open this way and the bottom part of the skull to open this way. And so we get a really wide opening of the mouth. Lots of movable bones within the maxilla and the palate. The spine is extremely flexible. We have a presacral, sacral, and caudal vertebral region. There is a single occipital condyle that forms the articulation between the skull and the spine, which allows a greater movement of the head. We have well-developed ribs, with the exception of the Chelonian. The turtles and tortoises' spine and ribs are fused to that bony shell. Skulls of the anurines are broad and fenestrated. They're holy, okay? Parts of the skull involved with olfaction and hearing are well developed, so they can smell and hear really well. They don't think so well. The palate is poorly developed with reduced dentition, but they do have some teeth. The caecilians have compact, well-ossified skulls with well-developed dentition. Caecilians generally live under surfaces, and so they do need to have a compact skull. Their musculoskeletal system, the amphibians have highly variable number of vertebrae depending on species. The vertebrae have no distinct regions. Salamanders and anurans also possess sacral vertebrae. Anurans vertebrae are fused, so that's part of the reason they hop. The urostyle is the last caudal vertebrae. Caecilians lack a sternum, and the sternum is a small cartilaginous plate in salamanders. Tail autonomy. This is a defensive mechanism in some lizards and salamanders. So they will lose their tail and it will continue to wiggle and distract the predator as they get away. So there are fracture planes within that tail along which the tails can break with little to no blood loss. That last tail can regenerate, but that regenerated tail is very stiff and cartilaginous, so a lot of scar tissue. The reptile appendicular skeleton, they have a pectoral girdle in the chest. It consists of the scapula and coracoid bone with muscular attachments to the body. The pectoral limbs are the front limbs that are the humerus, the radius, ulna, carpal bones, metacarpal bones, and phalanges, just like our mammalian pectoral limbs. The pelvic limb are the femur, tibia, tarsal and metatarsal bones, and the phalanges. You didn't see fibula in that. Okay, there's no fibula there. There's usually 5 digits on both the front and rear feet, just like us. Spurs are those vestigial pelvic limbs in some snake species, and they're bigger in males than in females. They're found on either side of the vent and are used in courtship behavior. Some lizards have very reduced limbs. Legless lizards actually retain a vestigial thoracic and pelvic limb structure. The pectoral girdle is primarily cartilaginous. These consist of precoracoid, coracoid, and the scapula. An exception to the amphibian pectoral girdle will be the anuran's pectoral girdle is completely ossified and consists of scapula, clavicle, and coracoid. The pectoral limb is similar to the reptile, except for radial ulna is fused in anurans. So that the frogs and toads have a fused radial ulna. There are 4 toes usually present on the pectoral limbs of the amphibians. The pelvic girdle has an ilium, ischium, pubis, just like we have with mammals. The pelvic limb is typically elongated and well developed for swimming and jumping with amphibians, and we have elongated metatarsal bones in anurans, usually with 5 toes. In 4-limbed amphibians and reptiles, the musculature is somewhat analogous to that of mammals, so we don't really go over that. With snakes, they have epaxial muscles and segmental muscles. And there are different ways that snakes can move. We have a concertina type movement. We have lateral undulation. It just depends on how they push off the surface of the earth. This sidewinder is moving to its right or its left. That's how it's directing its movement. It's not thinking of it as right or left, it's thinking of it as forward. And then there's rectilinear. That's what I have for reptiles and amphibians. If you have any questions, please bring them to me. As I said, diseases of reptiles and amphibians usually revolve around poor environment and poor education of the client. So let's discuss this in class. Make sure you write down your questions.